So in this last portion of our webcasts on protein structure and folding, we're going to finish off by talking about what's called quaternary structure. Now, compared to primary through tertiary structure, quaternary is actually the easiest of the batch to understand. All quaternary structure means is when you have the aggregation, i.e. the coming together of two or more protein chains, and we're going to term each individual polypeptide chain a subunit. The reason it's the easiest probably out of the batch to understand is because really quaternary structure is basically driven by the same forces that govern how tertiary structure forms, except now in quaternary structure we just have more than one polypeptide chain coming together via things like the hydrophobic effect or hydrogen bonding, what have you. Just to show you one example of how quaternary structure looks, we're going to use hemoglobin. Hemoglobin is the example that I think every single biochemistry book on the face of the planet uses. And it's the one I'll lead off with here first as well. When we look at the structure of hemoglobin, hemoglobin is basically made of four different polypeptide chains, four polypeptide subunits that come together and form this overall folded structure. Hemoglobin is referred to as a tetramer because it's made of four polypeptide subunits that come together. And it's actually called an alpha-2 beta-2 tetramer because it has pairs of identical subunits. So we have the two yellow monomers that have exactly the same amino acid sequence. And then you have the two burgundy subunits that have exactly the amino acid sequence. So this entire protein is simply the aggregation of four individual subunits. Later on, we're going to find out that when subunits come together to make the quaternary structure, a lot of times the function of the monomer is similar, but not exactly the same as it is in the overall tetramer. So in this example, hemoglobin can effectively be thought of as four different myoglobin subunits that have come together. Myoglobin is an oxygen-binding protein, but it's not what we call a reversible allosteric oxygen-binding protein. With hemoglobin, it turns out that hemoglobin has the ability to bind oxygen in a reversible manner, and in a manner where when oxygen binds in one subunit, it helps to change the affinity for oxygen binding in the other subunits. So it can respond very quickly and readily to changing in conditions to be able to either increase the picking up of oxygen or increase the rate or increase the delivery of oxygen. In contrast, myoglobin is either going to bind it really well or let go really easily. There's no sort of middle ground. So we're going to see more about these subtleties later on. But the idea is hemoglobin does have quaternary structure because it's made of more than one polypeptide chain, whereas somebody like myoglobin, which is only made of one polypeptide chain, by definition can only have up to or up through tertiary structure. Just to show you a couple more examples why I think quaternary structure is cool. What's really neat about quaternary structure a lot of times is that in a lot of proteins, it's simply many, many polypeptides folding together in a particular way. So on the left here, we have a molecule called ferritin, which is important for iron trafficking in cells. And on the right, we actually have a viral particle that kind of shows how quaternary structure looks. But the most important thing to appreciate out of quaternary structure in this slide is that a lot of times when you think about quaternary structure, the molecules have a lot of symmetry about them. And what I mean there is that the symmetry is important for how the job of the protein works. So these individual subunits work together to perform a function, and they have a very beautiful symmetry associated with them so that the molecule has a very logical arrangement of all of the different polypeptides. This will be really important for the function of some of the enzymes that we'll be talking about. So these are just some pretty examples of cool bits of quaternary structure. And so again, when we think about these guys, subunits are usually going to be related to each other's space by rotational or helical symmetry. So that is to say, you can, in a lot of these guys, if you simply do a rotation, if you imagine, if I said, you know, look at the yellow subunit, close your eyes and reopen them, you would, a lot of times, if there's rotational symmetry, you wouldn't be able to distinguish between The original picture and the final picture, unless I had things color-coded, because they fold together in a very predictive, symmetrical kind of way.